Hemostasis is the process by which we stop bleeding. Platelets is part of hemostasis. Hemostasis have 3 steps, and platelets play a role in hemostasis, but it's only one of the steps in hemostasis. So let's take a look at the platelets. We have in the bone marrow stem cell, different set of steps. We are going to get a cell called a megakaryocyte. A megakaryocyte is a big cell, and this cell is going to break into pieces, and the pieces are going to be the platelets. Imagine that the megakaryocyte is a cookie. The platelets will be cookie crumbs. Okay, so let's take a look at the characteristics of a platelet, and as you can see, it's these big cells right here. This is a megakaryocyte, and these ones right here are going to be the platelets. As you can see here, the megakaryocytes have a whole bunch of cell organelles, it has granules. You can see again megakaryocyte, and then here you see the platelets. So what's the function of the platelets? The platelets are going to form something called the platelet plug. Not only that, the platelets are going to secrete some chemicals that are going to produce vasoconstriction in the blood vessel. Another very important function is that the platelets also produce clotting factors, which are very important because they produce coagulation. They all have other functions, such as, for example, release chemicals that dissolve the blood clot once the blood vessel that is cut is already healed. Platelet production, we said that the platelet production happens at the level of the bone marrow by a process called thrombopoiesis. So let's go to the next step then, hemostasis. We said hemostasis has 3 steps, and as you can see here, vascular spasm is step number 1. Vascular spasm or vasoconstriction is the same thing. Platelet plug formation is number 2. And blood clotting or coagulation is step number 3. As we mentioned before, the platelets play an important role in all 3. Step number 1, we said vascular spasm. That's the step in which the muscle available in the blood vessels are going to produce vasoconstriction. Platelet plug formation. When the blood vessel gets damaged, areas inside the blood vessel are going to be exposed. When they get exposed, collagen is going to get exposed. And this collagen is the one that is going to trigger the addition, the attachment of the platelets to the walls of the blood vessel. Very important step is the presence of a substance called von Willebrand factor. Von Willebrand factor is like a glue that will help these platelets attach to the blood vessel to initiate the platelet plug. So once you have the platelets and the platelets start attaching to the blood vessel, the platelets are going to start releasing some chemicals by a process called degranulation, as you can see here. The platelets are going to release, for example, serotonin. Serotonin also will produce more vasoconstriction. It also has ADP. ADP will attract more platelets to the area that was damaged. And also thromboxane A2, which will promote platelet aggregation, which is the attachment of more platelets to the initial set of platelets that were attached to the blood vessel. The platelet plug is not very strong. So let's take a look what happens after you form the platelet plug. In this case, we're going to look at the steps of coagulation. Coagulation is achieved because in your blood you have coagulating factors. These coagulating factors get activated 2 ways. One way is the intrinsic mechanism, and the other one is the extrinsic mechanism. It's called intrinsic mechanism because it uses the clotting factors available in your blood. The other one is called extrinsic because the blood vessel opens. So factors in your blood vessels, after they get broken, initiate the coagulation. So that means that there are times in which your blood vessel is not damaged, but since we have coagulating factors in the blood itself, you can initiate coagulation even though you are not bleeding, or at least you're not seeing the blood coming out of your body. So let's take a look now at how we are going to use this mechanism. So you have extrinsic and intrinsic. Now there are multiple different factors, and these factors are activated in sequence, as you can see right here. One very important thing that you need to know is that the end result of the coagulation is that after you activate all these factors, you will activate thrombin, and thrombin will activate fibrin. The fibrin are going to be the fibers that will make this wall very strong. So you have the platelets, which are going to make the wall, and you have the cement that is going to be the fibrin. So the bottom line of all this, once you activate the coagulating factors by the extrinsic or intrinsic mechanism, you don't need to know the sequence, you don't need to know which ones are these factors, but you need to know that the end result of it is that you will activate thrombin and thrombin will activate or will produce fibrin. The fibrin are going to be fibers that eventually are going to produce this very strong mesh that we have in there that will help us stop bleeding.